Welcome back to Finding the Cause podcast. I'm Caitlin Ellis. I am the Executive Director of Find the Cause Breast Cancer Foundation and one of the 90% of women who are diagnosed with breast cancer with no family history of the disease. Find the Cause Breast Cancer Foundation is dedicated to all things prevention. Today, I am so delighted to have one of our consortium researchers with us, Dr. Charlotte Kuperwasser. Hi, Charlotte.
Hi, Caitlin. Thanks for the opportunity to talk with you today.
Well, I'm so glad you're here. So a little bit about Charlotte. I'm going to get through your title. I'm actually going to read it so I don't leave anything out. Dr. Charlotte Kuperwasser is the director of the Tufts Convergence Laboratory of Biomedical, Physical, and Engineering Sciences and professor of developmental, molecular, and chemical biology at the Tufts University School of Medicine. And she's a mom. So welcome, Charlotte. Charlotte, for those that aren't familiar with you or your background, can you just talk a little bit about your background?
Sure. So I've been working in breast cancer for about 30 years. Dates back all the way to actually my undergraduate experience, where I was actually working in a laboratory that actually introduced me to this field. It was a mammary gland biology lab. They were just studying the basic biology of how breast tissue develops. And I kind of fell in love with the model system and just the whole biology of the tissue. And so my background is I got my bachelor's degree in biochemistry in that lab. And then I actually stayed on and did my PhD in molecular and cellular biology at UMass Amherst. I moved on to MIT for my postdoctoral training in the laboratory of Bob Weinberg, a world-renowned cancer biologist, to really start to understand a bit more and dig more deeply into cancer biology. And then I moved to Tufts Medical School, where I've had my lab for about 22 years there and been working on breast cancer ever since. So really, with a lot of training on the molecular biology, basic biology, developmental biology, and cancer biology, that I think is a really great foundation on which I can actually continue to sort of study the intersection of all these different fields and how that can actually bring insights into the origins of cancer, obviously the treatment of cancer, but where my heart and my passion has always kind of relied on has been the prevention of cancer.
That's so interesting, and what a long career you've had, and we're so grateful to have you as part of the consortium. So talking about your lab, can you give us a little overview of your lab and how your lab works with the other labs? I know that's a lot to ask.
Yeah, my lab is part of the consortium that's funded by Find the Cause, an absolutely incredible organization that has just been not only a lifeline for cancer research, but really kind of pioneers in championing cancer prevention research. And so it's part of the consortium. My lab is part of that group. There's, I believe, 5 other labs together. We all kind of work on different pieces of this puzzle, which is, understanding and defining the causes of cancer, and then ultimately trying to find ways to prevent it. But before that and independently of that, my lab also works on other areas of breast cancer research. Primarily, we're really interested in understanding the cellular origins of cancer, some of the molecular and cellular biology, and sort of the basis of normal development and how that kind of intersects with cancer. We're also interested in the microenvironment and immunological factors that influence cancer. So we have a very large umbrella of breast cancer research. And then sort of Find the Cause Consortium is sort of one of those pillars in my lab that we focus on, but it's not the only focus of the lab.
No, that's great. And so talk a little bit about the collaboration of the consortium labs and back to those puzzle pieces. Who do you work the most closely with? Who do you hand off information, and where do you get your information when you're collaborating?
It's a great question. And what's actually really important and interesting about this is that it's not just one lab all the time. So I kind of view, and I think we kind of view the consortium as kind of a wheel where each of us has like an interesting area of focus, and then we actually collaborate with the various domain experts and labs in the other group to be able to kind of fill in the gaps where we don't have it. So for right now, one of the projects that I'm working on, or the lab is working on, is really tightly collaborating with Stefano's lab. So his laboratory, he's a pioneer in and an expert in computational biology. And so some of the work we've been doing has been really dependent and reliant on his expertise in being able to look at very large data sets that we collect and analyze them in order to make sense of that. And so for right now, we're working very closely with Stefano. But, 2 years ago, we were working very closely with David Scher when we were actually working on an immunology related project and trying to understand how the immune system in his animal model might be contributing to cancer. And then now moving forward, we're actually going to be working very closely with Jesslyn and Vicky. Vicky is a clinician out in California who has just been, dedicating her life to treating patients, particularly high-risk breast cancer patients. And so she has a wealth of knowledge on the clinical side, but she's also been following patient cohorts over her 30 years in the field. And so she has a very rich data set and understanding of these women over 30 years and their journey with cancer. And so working with her and Jesslyn, we'll be able to actually answer some of the questions we're interested in from their areas of expertise. So really, it's kind of a dynamic, evolving collaboration. The group, we meet actually every couple of weeks, and we all kind of go around and talk about the work we're doing and looking for synergies, looking for collaborations. And then when something comes up that's just a natural, obvious extension of our own research, we then cross-fertilize and synergize to move the projects forward.
No, I love hearing about the collaboration, and we will have each of those members of the consortium on the podcast to talk specifically about their work. Getting back to your lab, so tell me what's exciting. Like when you go to work every day, you tell me what are the things that excite you the most and what you see there on the horizon that's so cool.
Yeah, so I would say there's a couple things that's really exciting. One obviously is, and it's not my lab specific. It's more about sort of a system-wide policy and sort of appetite change that I think is exciting, which is historically cancer prevention research and even sort of understanding the causes of cancer have been underfunded. Just kind of a footnote in sort of the budget of the NIH, and kind of just sort of a cursory type of, there's something out there, but no one really is interested in studying it. And it has become front and center. And there's a lot of excitement there because for the first time in my 30 years, there's a real appetite to try to really understand what is causing this, unprecedented surge in cancers, particularly early onset breast cancers, and then trying to find a way to stop it and prevent it. And so I'm really excited, and I think that's really exciting because there's just been, I think, a societal shift, and that energy I find very exciting because it's not an uphill battle anymore. Everyone gets it. Like, we need to start funding this. We need to understand it. Therefore, so we have to stop it. On a laboratory level, on an experimental level, where I think things are really exciting has been sort of this new wave of technological advances in the laboratory in a way that we can study these questions in a faster, more cheaply, and more efficient manner than ever before. And what I mean by that is historically, studying questions that involve environmental exposures and cancer have required animal models. There was really no way around it. You basically had to expose an animal to a chemical and then look to see, does it develop cancer and what kind of cancer? And this was very painstaking. This was very time consuming. It was very expensive. It was one chemical at a time, large animal studies, and just very, very incremental. And so what's happened now that I find very exciting is we've moved to this new technology that's referred to as organoids, if you've heard about it. Essentially, it's a way that we can grow miniature tissues, miniature organs in the laboratory. So my lab uses breast, obviously. And so what this now allows us to do is being able to actually, recapitulate or recreate those experiments where you expose a tissue to a chemical, You look to see how that tissue responds, and does it respond in a way that actually primes it, to form cancer? Does it form cancer? And so we can ask these questions really quickly at scale in a way that we've never been able to do before. And that's really exciting because we'll be able to not only screen a lot of chemicals, but we'll be able to really rapidly translate those findings into actionable events, whether that's a change in policy, banning the chemicals, just being able to intervene in a way I think that can actually make a difference in our lifetime, not like, next generation,
Right.
even though I think that's what we're going for. So I find that really exciting. So we've got this new technology to be able to screen these chemicals. And now we also have, obviously, everyone knows AI has just been, helping to accelerate research in unprecedented ways. We now have these innovations that can actually help with the large data that actually comes out of these new scalable technologies. And so with computational, help, we have a lot of data out there that can now be mined and examined and scrutinized in ways that was never before possible, to also help us understand and identify causal and effect relationships between exposure and either, susceptibility to cancer or, in fact, full-on transformation to cancer. So that's what I find really exciting. That's where I think the new frontier is with our research, or in research in general.
Yeah, no, and so to just take that kind of piece by piece, you think about moving from an animal model over to being able to grow these organoids, right?
Yeah.
But being able to do that from a 10,000 foot view, what does that look like on a continuum? So how long does it take you to grow something that you can actually introduce to a carcinogen and actually then watch that and see what the changes are and when to intervene?
Great question. So it depends on the tissue, the organoid that you're looking at. So what's really interesting about these models is they tend to reflect the biology and the cellular turnover and growth of the tissue. In the context of breast, which is what we work on, there's kind of two approaches. There's what we call the developmental approach, or developmental organoid, where we start with a single cell, like a single breast cell, and we wait for that single breast cell to grow into kind of the basic tissue, the small functional tissue. That takes about 3 to 4 weeks. And so we actually then have an opportunity with that model to actually study the effect of the carcinogen or the chemical on early development. And so what that means in biology terms is basically fetal development and prepubertal development. So this is essentially when during pregnancy, when you go from the single cell all the way to the fetus, you can actually model the way the breast is growing during kind of that fetal growth. And then the breast also then develops in the second stage during puberty. And so these organoids, we can actually study the effect of chemical exposure from, in utero, so during pregnancy, all the way up until puberty. We have that can be modeled and rapidly accelerated because we can study this in 4 weeks, right? So, and we can watch it grow over that 4-week period. And so then we can actually treat those tissues right from the beginning to kind of mimic chronic exposure. So like a pregnant woman who's drinking her bisphenol containing water every single day, we can model what that's going to do to breast tissue as it's growing. Or similar, we can model that early adolescent girl drinking her water or her, contaminant, and we can see what effects it has on the tissue within that model. We have another model where we don't start tissue growth at a single cell. We start it as a smaller cluster of cells. And so there we're modeling the tissue in a later stage of development and in sort of a kind of what we call post-pubertal development. And so there we can also treat those cultures with the chemical and look to see how it affects growth those tissues, those organoids only take about 1 to 2 weeks to form. And so it's a much faster model, but it won't necessarily give us the same information as if we start with a single cell, which models, right, early fetal growth and early breast development.
Oh, I
Did
think
that
that's
make sense?
fascinating.
Did that make
Yes,
sense?
no,
Okay.
great
Okay.
explanation.
Okay.
And so when you talk about uh, being exposed to chemicals like the plastic in a water bottle, those kinds of things, and you think about prepubescent girls going around with big water bottles and from the store and plastic straws and those kinds of things. What is the mechanism that you're looking for there? What happens there that would be interruptive later in life?
So that's the $1 million question, right? So this is exactly the gap of knowledge that we're confronted with today. So we can look at sort of historical data. So we know that certain chemicals will affect, and these chemicals, they're endocrine disrupting chemicals. So this is a very large umbrella of chemicals that encompass a wide variety of chemicals and exposure routes. And so these are chemicals that are found in nonstick cookware, in the food packaging, they're in water repellent fabrics, they're in the air, they're in your cosmetic, they're in your your sunscreen, nail polish, dental floss. You were just saying the plastic water bottles. They line the aluminum cans and the soup cans. They're flame retardants. I mean, the list goes on and on and on. There's thousands and thousands of these, and they're called endocrine disruptors. And the name, and it's important to say because, endocrine disruptors, everyone thinks they're just like your bisphenols, the things that we hear about. But these are chemicals that have a lot of different chemistry. But what they all have in common is that the EPA, when they look at these chemicals, they can show that these chemicals interact with hormone receptors in the body. And the hormone receptors are, primarily the estrogen receptor, the progesterone receptor, the thyroid receptor, the androgen receptor. Those are kind of your big guys. There are obviously other receptors. I can go through the list, but those are kind of your big ones. And they're big because they influence so many different parts of the biology and physiology. And so what we focus on are really the ones that primarily affect the estrogen receptor, the progesterone receptor, the thyroid receptor, because those are kind of the big three for breast tissue. I mean, everyone knows estrogen and progesterone, but thyroid also plays an important role. Those are your big three ones. What they do is a lot of different things. They bind to the hormones, right? That's their definition. But their downstream effects are very varied, and ultimately what that results in with respect to changes in breast tissue, we don't fully know, right? And that's the gap we're trying to understand, is if the breast tissue is exposed to one of these endocrine disruptors, does anything happen, right? I think that's even the first question we have, right? We have, again, some clues based on bisphenols. So we have a lot of and primarily bisphenol A. I should clarify that. Not all the bisphenols have been studied, but bisphenol A, we have a lot of information on that. We know from animal studies that exposure to bisphenol A does disrupt breast tissue development in a way that allows it to become susceptible to cancer later on. And that's in part why it was banned from baby products and bottles and plasticware. But the unfortunate reality is it was replaced with what we call the regrettable substitutes, and so it was replaced with bisphenol S and bisphenol F. They just stick another letter to it. It's just pretty much the same chemical with a modification. And there we're only starting to study and understand what those effects are. And we found in the lab and in our studies that it actually does disrupt breast tissue in a very similar way that bisphenol A does. So basically all we've done is basically just moved moved the goalpost, but it's the same goalpost. We've gone from one bad bisphenol to another bad bisphenol, and so those are still out in our environment, right? So the downstream mechanisms yet are still being worked out, and that's, part of what, this foundation and what the work that we're doing as a consortium is trying to understand. The other gap we don't actually know and haven't been able to show, I think, until recently, is whether exposure to those chemicals does in fact lead to cancer or have any relationship with cancer, right? So we have the experiments where in animals there's disruption in breast tissue, but does that actually ultimately lead to cancer or change susceptibility to cancer? Is there a fingerprint of that exposure in cancer? Because that's kind of what you need. You need to be able to show that cancers might have resulted from that prior exposure. And some of the work that we've been doing has actually started to give us insights into the answer to that is in fact yes. That exposure to these chemicals early on, you can find evidence of that exposure in the tissue right next to the cancer. And so it's kind of a smoking gun to telling us that these exposures have likely a very strong influence on cancer. The other example that is commonly used for how we know that endocrine disruptors cause cancer is actually historical work from women who were given a chemical called DES during pregnancy. It turned out that the daughters of these women actually ended up developing a rare form of cancer, reproductive cancers. And that was actually kind of the first clue that exposure to these endocrine disrupting chemicals can cause cancer. The mechanism of that, interestingly, is still not known. 50 years later, we don't actually know precisely the mechanism by which exposure to that chemical during pregnancy caused cancer in their daughters. But it does point to a mechanism that involves remodeling of the DNA and changing of the tissue developmental program. And so we're seeing similar type of outputs or similar types of results when we treat breast tissues in the dish with these endocrine disrupting chemicals. So ultimately, while the underlying, specific mechanism might be varied from chemical to chemical, the output is you have a tissue that has been primed, that has been altered and changed in a way that makes it very susceptible for transformation in cancer much earlier on in a person's life than historically.
I think that's so fascinating. And in talking about identifying those parts of primary prevention, the things that you can avoid in your everyday life that we're identifying that cause these endocrine disruptions. In your career, I mean, if you've been doing this for 3 decades or now, and that's incredible. Over the years, tell me a thing or two that you've adapted that you suddenly said, I'm not using that anymore, or I don't want, it's just, give me 1 or 2 things that you use and 1 or 2 things that you'd share out that the people listening today could do to really control their environment and lower their risk for exposure.
Yeah, that's a great question. And first is I don't want to be alarmist. We're all kind of in this together. We are in a time where we are bathed in chemicals, and I became extraordinarily neurotic, and I still am, because when I was pregnant with my daughter, and even now. So I will give you what a high level of what I do. I can't just specifically say 2 things because I do like 100 things, and again, it's neurotic. But what the advice that I kind of tell myself when I'm constantly being like, okay, I need to just like relax a little bit, is you basically want to identify what I call sort of the big ticket exposures. And big ticket exposures are basically any type of exposure that you're doing every day, period, right? And so that usually falls into what you eat, what you drink, and what you put on your skin, right? So those are what I call the big ticket exposures. And so what I've done the 2 things is my source of water is really, really strict. I do not drink tap water. I have a filter on my tap water. And then I also sometimes, so I have kind of two filters. I have an RO filter and then I have a secondary filter. So again, a little neurotic, but water is a big one. I
And tell
try to
people what
avoid.
RO is. What's RO?
Oh, RO is reverse osmosis. So it's basically a filter you can buy. You can put it under your sink. It basically runs it through filters and basically cleans everything out. But you have to put the minerals back into your water because it strips the minerals out too, and you can't be drinking unmineralized water. But it basically strips it out. The other way you could do is get yourself a distiller, distill your water, again, then put the drops back in. But you want to find a way to make sure that you're cleaning your water because as much as, we hear that your tap water is fine, it is not. I mean, there's PFAS, there's all sorts of chemicals that are in there. They might be within the healthy limit, but again, we're talking about cumulative exposures and we're talking about combined exposures. And so if it was just PFAS in the limit in your water, fine. But that's not what the reality is. It's PFAS plus, 800 other chemicals you're exposed to every day. So the goal is you're just trying to kind of limit the big ones and you're trying to limit your cumulative and chronic exposure as much as you can. So water is my biggie. I also don't have plastic water containers. I have glass water containers. So that's kind of a biggie. The other biggie is relying, again, it's based on food, right? Food and water, the big ones. The other biggie is my food containers in my house. Everything is glass. I do not own any plastic Tupperware, and I never microwave anything in Tupperware. And if I buy frozen meals that come in that black plastic Tupperware, I dump it into a glass container and then I microwave it. Microwaving plastic is a really dangerous way to expose yourself to food contaminants. And so that's kind of a biggie. And then another really big one, and there's going to probably be a lot of backlash and controversy, but it has to do with lotions and sunscreen that go on my body or my daughter's body. So I've stopped with just about every possible brand. Sunscreen, I only use mineral-based sunscreens. Yes, it's white. Yes, it's hideous. But until they've cleaned up their products, sunscreen is really full of endocrine disruptors, as are a lot of lotions. So I make sure I read the ingredients, and so I'm very particular about the brands of cosmetics and skincare that I put on my body because again. Every day, right? So these are the everyday exposures, and so you just want to minimize that chronic accumulation. But yeah, I mean, the list can go on and on and on, but I feel like those are the big ones. If you want to then do kind of the secondaries for me are the cookware, right? But I've already cleaned those out. So I use stainless steel, but and you're cleaning products and things like that. But if you're not cleaning every single day and exposing yourself every single day, it's not as, right. I mean, you got to pay attention, but it's just, again, it's the big ticket ones, the daily exposures, the chronic exposures, then the cumulative. And because they're mixed and you're constantly getting it from all sources, you just want to try to minimize the major ones.
And I think that's a big thing with the Find the Cause education program. A lot of people think, oh, a small change I can do is to store my food in glass containers and not plastic. And to tell you the truth, my strawberries last a lot longer in a glass mason jar than they do in plastic Tupperware. So I'm all for that. But so thinking about what would it mean to you as a mom, as a researcher, to get to the end goal of breast cancer prevention? I mean, that the actual, lock the door, turn off the lights, we did it. What would that mean to you?
What would it mean to me, or how would I kind of measure that we've done it?
Oh, how about both?
Okay. Well, so because I've been giving this a lot of thought because, I realistically, we can't prevent all cancers or cancers in general. Cancer is part of life. Cancer has always been part of life. Um, you know, after the age of 40, probably even before now, we're all walking around with cancer cells. We can't kind of prevent cancer because this just is part of life. It's just part of, you know, just the natural process of living, of cell turnover, of cell division, of aging. So my view is the goal is not to kind of prevent cancer, because I just don't think that's possible. My goal is to basically make sure that no one dies of a cancer that we could have kind of prevented from making it life-threatening. And where I actually really think that, where I would say, okay, I'm going to shut down, close it up, and call it a day, is if we can prevent cancers in anyone under the age of 40. So but after the age of 40, again, I don't think we have a way of preventing it. We can just prevent it from, you know, manifesting into a life threatening chronic disease. But I don't think we can really prevent it. It's just, but under 40, there should be no one who's getting cancer under 40, as far as I'm concerned. And, you know, one can say, well, what about the genetic susceptibilities? And I would say, well, we've gotten really good at screening. We've gotten really good at identifying genetic families. And so, again, by early monitoring, they will not develop, they should not be developing a life-threatening condition or disease from it. But that's you know, we're not seeing that right now in the general population, where it's an epidemic of under 40s that are developing breast cancers. We've got 20-year-old girls who are developing breast cancer. Like, this is unacceptable. And so for me,
and very aggressive forms of it too.
For sure, exactly. And so, and it's within the last 30 to 40 years that we're seeing this. And so this is like unacceptable. So for me, stopping that, then I'll close up shop and retire. And so that's sort of how I would gauge it. What it would mean to me, honestly, it would make me feel that we've contributed an impactful and meaningful quality of life for the next generation, for my children, for my daughter. You know, I do think about, and I'm sure all moms think about, you know, what their daughters are going to grow up to be in their lives. And the last thing I would want is to imagine that she developed breast cancer. There's no reason for it. We have no family history, and so if she does, that's a failure. So that's really kind of what I just think that would be what we're working towards. You know, there's no reason in this day and age, with the technology, the know-how, the knowledge, the infrastructure, and hopefully the financial support. I mean, again, you know, with what you guys are doing, absolutely, but we need a larger support here. This shouldn't be happening. We should not be having, you know, teenagers, 20-year-olds, 30-year-olds, with no family history, with no reason to suspect anything, having cancer. So, I've already forgotten the question, Caitlin. What was the question?
I love it. Well, you nailed it.
I don't remember.
You nailed it. What would it mean to you, and what does that look like?
Okay.
Well, Charlotte, I appreciate so much your time today, and I invite you back when we start to pair up some of the researchers whose collaboration within the consortium work together to help illustrate what that looks like. One last question that I'm going to ask is that, tell me what the work of Find the Cause means to the research that you're doing. How has Find the Cause and the supporters of Find the Cause influenced the results in your lab and kind of the progression there?
Yeah, so that's a really great question. So, like I said, I've had my lab for 22 years, and we've been working on many different facets and many different directions in breast cancer research. And I would say when Find the Cause came to me and kind of explained to me their mission, that actually caused an actual pivot in the research program of my lab. And so what it's meant to me is that we were actually able to start to really study the fundamental question of what is the cause of the breast cancer in a way that we've never been able to before, because there was no support. And so because of it, we now have a number of studies and a number of papers and research that have come out and have just been directly financed by Find the Cause. I mean, there's been no other funding agency other than Find the Cause that has supported this work. We have a paper that's going to be coming out very soon, 100% funded by Find the Cause, the only group that was actually courageous enough and supportive enough for this work, where we've now directly shown that exposure to endocrine disrupting chemicals affects and alters breast development in a way that we can now see it in patients, we can see it in normal tissues of women, and we can actually see that it's leading to the development of invasive lobular carcinoma. So it's basically taken the entire journey of cause to effect, completely funded by Find the Cause. And so what it's meant to my lab is that we are now able to, have a really solid, strong pillar of funding and focus on this problem. And we're making direct impacts into it. And hopefully that this work will actually prompt either policy changes or at least some type of larger scale effort to really assess chemical exposures and the type of breast cancer and the rate of breast cancer and the susceptibility of breast cancer. So it's meant not only changing the focus of my lab, but also supporting it in ways that no other agency had the courage to do, and still doesn't have the courage to do, but hopefully it'll change.
Well, I think that's so great for all of our supporters to hear. And of course, on Find the Cause's website, which is findthecausebcf.org, we link to all of your published papers as well as the rest of the consortium. So if anybody wants to dig a little deeper, we certainly have all that information there. Charlotte, thank you again for your time today. I am so grateful to work with you, and your enthusiasm and your passion around prevention is clearly contagious. So thank you so much.
Thanks, Caitlin. This was really great. Glad I could participate and help and spread the word.
Thanks so much to Dr. Kuperwasser, and I appreciate you joining us today. You can find this episode and the previous episodes anywhere you find your podcasts. So stay tuned for the next Finding the Cause.